So inside the sarcomere, there are both thick and thin filaments, along with other associated proteins. Let's take a moment now and talk about the thick filament and how it's structured. We said earlier that thick filaments are made out of myosin proteins. The thick filament is actually composed out of two bundles of myosin proteins, and there are actually hundreds of individual myosin molecules inside of each thick filament. The two bundles of protein are arranged in opposite directions, forming a mirror image. The heads of the successive myosin molecules are going to be arranged in that bundle, in each of those bundles, so that they protrude off of the surface of the bundle at right angles. And I'll show you a picture of this in just a minute. These bundles of myosin molecules are not randomly put together. They are arranged very particularly so that the heads jut off in a way that allows them to contact the thin filaments. Remember, the thin filaments are arranged around the thick filament in 360 degrees. That hexagonal pattern matters. It's important. The globular heads have to be able to interact with the individual thin filaments that surround the thick filament. Everywhere that the protruding heads can come in contact with a thin filament, there's going to be a binding moment and a cross bridge is going to be formed. These cross bridges, these locations where myosin heads bind to thin filaments, they are temporary structures. So we're going to see binding occur, and then we're going to see a release. A cross bridge is going to form, and then a cross bridge is going to be broken. On this slide, you're looking at a diagram of a thick filament up at the top. And then the artist has taken a little region of that thick filament and blown it up for us so we can see it in detail. So if we start up at the top, this thick filament is actually made out of two bundles of myosin molecules. There's one bundle here, and there's one bundle here. And the two bundles are brought together and attached to each other right in the center. So these bundles are oriented in opposite directions so that there are heads protruding off on this end and heads protruding off on this end. Remember what each individual myosin looks like. There's that coil of heavy chain. That's what would be located here. And then the heads jut off at one end. Over here, we see the same thing, just oriented in another direction. Now these myosin molecules, they're staggered away from each other in the bundle so that the heads jut off of each individual myosin all the way around the circumference of the thick filament. The heads are oriented in what might appear to be a random pattern. But if you look closely, for example, if you look at the heads that are jutting off of the bottom of this region of the thick filament, they're actually oriented in very regular patterns. So there's one jutting off of the bottom right here, one pair of heads, and then there's a distance until we see another pair, and then that same distance until we see another pair, and so on down the length of the filament. This is happening all over the circumference of the filament. Heads jutting off in 360 degrees in a regular pattern. The thin filament looks very different. The thin filament is actually made out of two strands of actin molecules wound around each other. We've seen this type of structure before. Remember, we talked about microfilaments being made out of F-actin molecules, filamentous actin molecules, joined together in a linear strand, and then two strands being wound around each other to make up the microfilament. Two strands will be wound around each other to make up a thin filament as well. But there are going to be two very important proteins associated with those two strands of actin. There's a protein called troponin, and there's a protein called tropomyosin. When you look at a tropomyosin molecule, and I'll show you a picture of this in just a moment, it's a long, twisted, rod-shaped molecule, much smaller than the thin filament. It's going to be sitting at intervals along the length of the thin filament, oriented parallel to the thin filament. If you imagine the two strands of actin that are wound around each other in a thin filament, those two strands, where they come in contact with each other, are going to create a groove that twists around the circumference of the thin filament all along its length. The tropomyosin molecules are going to be sitting close to that groove. They're not in it, but they're lying very close to it, parallel to it. The groove is important because it's the groove along the length of a thin filament where the binding sites for the myosin heads are going to be located. Each of the tropomyosin molecules, as it lies along the length of the thin filament, it covers 7 actin monomer units. So if you think of a thin filament having many individual actins along its length, the tropomyosin molecule is much shorter. So there are many tropomyosins lying along the length of a thin filament. Now troponin looks very different from tropomyosin. There's one troponin for each tropomyosin on a thin filament, and the troponin is much smaller than the tropomyosin is. The troponin protein is actually made out of 3 individual polypeptides that come together and work as a unit. One of them is called TNT, one of them is called TNC, and one is called TNI. If we start with the TNI subunit, this is the part of the troponin molecule that binds to the actin in the thin filament. So troponin is attached to the actin in the thin filament. The TNC portion of troponin actually binds to calcium ion, and the TNT component binds to tropomyosin. So the tropomyosin is bound to the troponin, and the troponin is bound to the actin. All 3 of these things are bound to each other to make up a thin filament. Let's take a look. In this drawing, we can see one thin filament, or at least one portion of a thin filament that comes out of a sarcomere. The blue circles represent individual actin molecules. We call this F-actin, remember, once it is polymerized like this. So we have 2 strands of F-actin that are wound around each other, very similar to what we saw in a microfilament. The difference here is that there are some additional proteins that are bound to this actin. There's this green protein. I often refer to this as looking like a licorice stick. It looks like a red vine or a licorice stick. Notice that it's lying parallel to the strands of actin, and it's lying close to the groove that is created between the 2 strands of actin. it's much shorter than the thin filament is. So you can put several of these green licorice sticks down the length of the thin filament. These are the tropomyosin molecules. Each individual tropomyosin is only covering 7 actin molecules. Now, the troponin you have to look very closely for. The troponin is located right here. There's one troponin for each of these tropomyosin licorice stick molecules. And each troponin has 3 regions, 3 individual polypeptides that come together and make the entirety of troponin. There's a TNI subunit, that's the part that binds to the actin. There's a TNC subunit, that's the part that binds calcium. And there's a TNT subunit, that's the part that binds to the tropomyosin. So the tropomyosin, the troponin, and the actin in the thin filament are all bound together. Now, one of the things you might ask when you look at a structure like a sarcomere is how in the world do those filaments stay so tightly organized? How in the world do you maintain that perfect hexagonal orientation of thin filaments around thick filaments? The answer is that in addition to the thin and thick filaments in a sarcomere, there are additional proteins that help maintain the architecture of the sarcomere. Remember, the thin filaments are anchored at the Z line. They're anchored so that they come off the Z line perpendicular to it. The thick filaments are anchored at the M line in the area where there are no globular heads. The thick filaments and the thin filaments have to stay parallel to each other because the thick filaments are going to bind to the thin filaments and slide them. Remember, the thick filaments contain the motor protein. The globular heads are going to reach up essentially and bind to the thin filaments, and then the globular heads are going to slide the thin filaments a short distance. The thin filaments are actually going to move toward the M line, and I'll show you a picture of this in a minute. What that means is that during contraction, the sarcomere is going to shorten, even though the thick and thin filaments themselves don't change length. The sarcomere is going to shorten because the thick filaments are going to work to slide the thin filaments towards each other in the center of the sarcomere. Now, I said that there are other proteins that are located in the sarcomere that just help maintain its architecture. I have a list of them here. I'm not going to hold you responsible to learn the names of these proteins. I just wanted to show some of them to you. There's a protein called CAPZ. There's one called tropomodulin, myomycin. There's a protein called titin. There's one called nebulin. These proteins are all found in the sarcomere, and they're located so that they maintain the architecture of the sarcomere. They keep these thick and thin filaments perfectly lined up with each other so that they can bind to each other in the region of the globular heads, and we can have a sliding motion occurring. So the globular heads that you see on this side of the thick filament are going to grab on to the thin filaments, and they're going to slide them this way towards the M line. The globular heads over here are going to grab onto the thin filaments on this side of the sarcomere and slide them this way towards the M line. So if we come back after contraction has been allowed to occur, the Z lines are going to be closer together. The sarcomere is going to shorten. not because the individual filaments have gotten any shorter, but because they've moved. The thick filaments have moved the thin filaments this way and this way towards the center of the sarcomere. What we're going to notice under the microscope as the sarcomere shortens. Remember, the Z lines are going to come closer together. The A bands are going to stay pretty much the way they look now. But these I band regions are going to get narrower and narrower and essentially are going to disappear. So the sarcomere Z lines will go this way until they bump into the edge of the A band. This one will move this way until it bumps into the edge of the A band. On this slide, we're looking at part of a sarcomere blown up so we can see more of the detail of the accessory proteins that help a sarcomere keep its architecture. So there's a Z line located here. Remember, the Z line is the place where the thin filaments are anchored. So you can see a thin filament here in blue. There's another one up here, and another one and another one. There would be thin filaments coming off of this side of the Z line as well, going into the next sarcomere. The thick filaments are shown in brown. And of course, they have globular heads coming off at regular intervals. Over here, we can see the other side of the thick filament, but we can't see any more thin filaments. They've been removed. So we're going to focus just on this region over here. You can see that they've drawn in a protein here that is connecting the two mirror image bundles of myosin in the thick filament. That's myomesin. Notice that there's a green protein right here. That's tropomodulin. It sort of caps the ends of the thin filaments. There's a protein called nebulin that sits over here. There's another one called titin, which sits between the end of the thick filament and the Z line. Titin is a really interesting protein, just as a side note. It got its name because it is the largest protein in the human body in terms of the individual number of amino acids. It folds up into a spring, and it functions like a spring. In other words, it's helical, and it sits in this location because it will prevent this thick filament from crashing into the Z line. Remember, this thick filament is going to slide this thin filament, these thin filaments, this way towards the center of the sarcomere. So this Z line is going to travel this way. And we wouldn't want the Z line and the end of the thick filament to crash into each other because they could damage each other. Titin prevents that. Titin acts like a spring. Notice that there's a purple protein drawn in here, sort of laying on top of the Z line. This is called cap Z. There are lots of individual proteins that are located inside of a sarcomere that serve as accessory proteins. They're not doing the primary work of the sarcomere, but they are helping the sarcomere keep its very precise architecture. On this slide, you're looking at a table that shows these individual proteins that are found, at least in vertebrates, and what their role is in the sarcomere in terms of helping it maintain its architecture. Remember, I don't need you to learn the names of these individual proteins. I just want you to know that there are a whole group of proteins in the sarcomere outside of the thick and thin filaments, and they're critical to the normal functioning of the sarcomere. I want to move forward now and start talking about how the sarcomere shortens, how the thick filaments slide the thin filaments towards the center of the sarcomere and pull those Z lines together so that the sarcomere shortens, so that the myofibril shortens, so that the muscle fiber shortens, so that the muscle itself contracts. This work, the work of determining how sarcomeres contract, was done many years ago, and the scientists who did this work gave this process a name. They called it the sliding filament model of contraction. This model has held up. We believe that this is the way that sarcomeres shorten, causing muscle contraction. The thin filaments are going to slide past the thick filaments with no change in length of either filament. This is a movement that's going on that slides the filaments past each other without actually shortening the filaments themselves. The A bands that can be seen under the microscope are going to stay the same. The I bands are going to get shorter and shorter until they disappear. The sarcomere is going to shorten until the thick filament ends come in contact with the Z lines. As sarcomeres get shorter, the whole myofibril gets shorter. And as all the myofibrils get shorter, the whole muscle fiber contracts. Remember, each muscle contains many, many muscle fibers. So as all of those fibers get shorter, as they contract, the whole muscle will contract.